interactions, but I'm interested primarily in the chemical and molecular mechanisms that mediate interactions between organisms. And so it's pretty much plant chemistry and how that plant chemistry functions as information in the world. I'm currently in Colombia on a sabbatic leave, so I'm totally relaxed, and I get this weird idea. So if anything comes up that is really weird what I'm saying today, it's because of that. And there is something about the landscape. This is an area in the Sierra Nevada de Santa Marta on the Caribbean coast, one of the most biodiverse places on Earth. And I'm coming from Germany, from an area where the Romantic poets come from. And at a site like this, I literally cry. And there is another thing that happens when you look at those forests. So this is a high Andean forest that Carlos is actually coming from, not from the forest really, but from near Bogota, where this is the image that I have and always had when reading the last pages of The Origin of Species, where Darwin says, a tangled bank clothed with many plants of many kinds, with birds singing in the bushes, with various insects flitting about, and with worms crawling through the damp earth, which have all been produced by laws acting around us and have been and are being evolved. What a wonderful sentence. It's actually several sentences kicked into one. But what's important about that, the view that he had there was a very beautiful one, very romantic one, makes me cry. But the interesting question is how those interacting organisms that drive each other's evolution interact ecologically with each other, and how those mediating factors, the interaction mediating factors, drive the evolution, in fact, or hinder the evolution. And these are some of the questions that, in fact, and this is what I want to talk about today, drive chemical ecology currently, but they can almost only be answered in tropical systems because there we have the diversity to do actually comparative studies within a similar habitat. And this is something that came to me in Colombia, where you can drive 30 kilometers and you're in a completely different habitat. You can be from tropical rainforest to a desert in the 30 kilometers, right? And so you have these fundamental differences on a landscape scale, but even on a small scale, you have incredibly high diversity within, for example, one plant family or one plant genus. I'm just talking plants here. With insects that would work too, and with any other organism group as well. Now, the thing is, though, when I step into, and I'm not sure how you kind of feel about that, when I step into a rainforest, for example, I'm overwhelmed by the beauty to begin with, but also overwhelmed by the task to even formalize the question appropriately and then to find enough organisms to do something on a population ecology and community ecology level. So if we leave alone tropical research that is focused on protecting those wonderful habitats, and we ask, what do we know and what do we study when we look at community and population biology in tropical systems, we end up getting stuck on the immense diversity that is there. So in many cases, although the best ideas came from tropical biology, they have actually been tested in much simpler, less complex systems in the temperate zone, right? And so we are kind of focusing on big questions about the generation, the maintenance, and the function of biodiversity and interaction complexity. However, get blocked by the fact that we cannot identify the organisms that we are actually working with, right? And Carlos gave a wonderful introduction because he's one of the few people who have really started diving into that and saying, I don't care because I can just say this is a species and that is a species by barcoding and don't care whether or not it has a name or not, right? But that way, it opens a door that is incredibly exciting and allows us finally to treat tropical systems very much like we used to do when used to work in the lab or in temperate zone simple systems. And there's even developments in technology that make it more and more easy. So there's this wonderful thing, where do I point to here, called the mini ion by the Oxford nanopore technology. So who was here at the Tropical Ecology meeting that was here in San Jose a few years ago, when Dan Jensen gave this talk about fingerprinting and genotyping, he said, and he took out his calm, in 10 years from now, the sequences will be this size. So here it is. It's even less than 10 years from then. This is a sequencer the size of a cell phone. And you can transport it wherever you want, and it seems to work reasonably well. It's not the best thing yet, but it will be there. The same in the field that I'm working in chemical ecology. There are now things like a portable GC that you can carry into the field, and you can sniff on plants and have results within seconds, in fact. So there's a GC that actually runs at an average 30 seconds long, and you can have high time resolution, and you can have a high throughput for ecological studies. So those technologies allow us to not ignore the diversity, but embrace the diversity and finally get to work on the big ecological questions that were kind of hindered by the immense diversity that we are facing. And we can look at diversity as such. So what I want to talk about is, in fact, the promises for ecology that are buried in that. And on one hand, the big ideas in chemical ecology, in fact, also came from the tropics and from people like Dan Jensen, for example, right? And the pioneers in chemical ecology like Tom Eisner and Jerry Meinwald. And Tom Eisner, for example, was a big proponent of the bioprospecting idea, but not through random screening of secondary metabolites or whatever toxins that come up in insects or snakes or plants. He was saying if we can understand the function of a particular compound in an organism in nature, we can derive a particular function for whatever our physiology in medicinal use or as a pesticide and things like that. So bioprospecting was a big thing in the 80s and 90s. Tom Eisner, in fact, his entire life had only one grant that was renewed over and over again over his entire career from NIH with that prospect, right? Nobody would get that anymore, and there's a reason for that. Not because the funding is down, but it's interesting that there's not much money, actually, for a pharmaceutical company to find an extract because you cannot patent the compounds. So it's much better to design a compound because you can make lots more money with that. So bioprospecting kind of came, even though it's still done, right, and has to be done, but there is not as much money there anymore for it. The other part that is important in chemical ecology, and this is what I actually want to focus on today, and what I forgot to mention, bioprospecting is the biggest thing actually in the tropics, right? Because here you have an unbelievable diversity of species, on top of which is an even larger diversity of secondary metabolites in those species, right? And so it has to be a focus for the future, but on the other hand, the interactions that are mediated by chemistry are those that frame the bigger questions in ecology and evolution. And there are things like mediated, and I just put this here to say that pretty much every type of interaction that you can define ecologically and any outcome of those interactions have been shown already to be somewhat, and to a certain extent, sometimes to very big extents, mediated by chemistry. And for my talk, I want to focus today on plant-herbivore interactions because this is what I know to talk about. And one thing that I need you to know to begin with. There was a big step when chemical ecology was started to understand that compounds or chemicals in nature can function as information that transfers is used by transmitting information between organisms. And this is just an example for plant-herbivore interaction. So the chemistry that a plant has, the totality of the chemistry, which is indicated here by that colorful ring around the plant, determines which organism can actually interact with that plant, right? You can evolve in and out of that ring, right? But ecologically, that ring, the totality of the secondary metabolites produced by the plant, determine who is there. And there are several different mechanisms how that happens. So on one hand, there are toxic compounds, very obvious. If you bite into it and you die, you shouldn't be in that circle, right? But there are interestingly also non-toxic cues associated with toxic deterrent compounds that simply are information that are good to be used by an organism who cannot handle the toxins. So they're information that advertise the toxicity of a plant and thereby can keep a certain number of organisms already out of that circle. And then there are very interesting compounds that are called attractant compounds that confuse the search patterns. So they are neither toxic, nor do they actually carry information, but they add noise. This is pretty much in the terms of communication or information theory. They add noise to the spectrum that the plant is producing. And we think that a lot of the diversity that we see in plant secondary metabolites is due to that fact, because you can, as a plant, evolve other organisms out of that ring by just adding a compound that confuses the search pattern of the herbivores associated with that plant. So in other words, the herbivores don't perceive that plant as a suitable host. Now, when you add the plant's ability, and this is important to understand for what I'm saying later, plants have the ability to change their metabolism in response to environmental stresses in general, but in particular in response to damage by herbivores, pathogens, and things like that. And what that does is it adds information about the physiological and metabolic status of the plant to all organisms that can interact with the plant. And we know that these changes in secondary metabolism, for example, can attract predators that then go to the plant and kill the herbivore that's on the plant. We call that indirect defense, right? It can give other herbivores the information that there are mates on the plant, and so it would be a negative effect in this case because the herbivores would accumulate on that plant. But on the other hand, herbivores can also avoid this plant because it's already damaged and has a higher defense status, right? And there are things, and I will give you an example about that too, where plants can communicate with each other. And here becomes very, very interesting because suddenly plants become a potential model for communication per se. And I will give you an example in a second. And I also will give you an example of where there is evidence for conflicting selection on those chemical information traits, namely when you have herbivores inducing, and this would be herbivore as an antagonist of the plant, right? Inducing a change that then screws up the interaction with a mutualist like a pollinator. And I'll give you an example in a second for that. So what we have there, in other words, is that there's a specific adjustment of the metabolism, as I mentioned, and which involves the entire, which then can involve the entire interaction network in a potential defense strategy of a plant. And this thought alone is a very interesting one, right? So if you think about a diffuse co-evolutionary process of a community of organisms with a particular population of plants, right? Then the plant could potentially evolve, and we don't really have too much evidence for that yet, using its entire interaction network. Here we go. So chemical ecology has recently undergone, is that a word? Has recently undergone a shift from being a complete mechanistic science to being much more functional and evolutionary. And there are certain bigger questions that colleagues of mine at Cornell and I summarized in this paper here in Ecology in 2012, which was about the future of chemical ecology. And it occurred to me in preparation of this talk today that everything we listed there is best studied in the tropics. And so there are things about the role of chemical signals in aiding, preventing diseases or pest outbreaks and affecting their dynamics. So this is something that's ongoing, has been done for quite a long time, and this will continue to be a big issue in the future. Things like that, though, open the question about, do organisms communicate with chemistry differently in different habitats? Very much like birds communicate differently vocally let's say in an open versus a closed landscape, right? Do particular communities select for different chemical communication among organisms? Are chemically mediated associated resistance and susceptibility something that's driving community structure? Then we talk about chemical signaling between multiple trophic levels as a determining factor for competitiveness and thus persistence in a particular community. I will give you an example for that. So the question is, if organisms share information about their metabolic status, and I'm talking from the plant's perspective right now, does that actually affect community dynamics? In other words, does it prevent or enhance the possibility of a particular outbreak, for example, or that particular organisms are interacting with the plant, right? And there is the potential for invasive species to, is more of a question, right? Is there a potential for invasive species to alter chemical interactions between organisms, leading to alternative stable states in the communication or pretty much the information landscape that's surrounding an organism? And this is the chemical information landscape surrounding an organism. Okay. I give you a few examples that kind of supposed to illustrate some of those questions that I put there, but I want you to keep in mind and kind of think always one step higher. So how would you kind of approach the kind of interesting natural history that I give you from an evolutionary perspective, and how would you study it? And you will see if you do that, it's best studied in the tropics because you can do comparative studies on a larger scale. So first, we go to Peru, where we studied wild tomato plants on the eastern, or the western slope of the Andes, so the Pacific slope of the Andes. And these are habitats like this. It's actually called seasonal triforest, but it's kind of more of a shrubland. There's only one big tree species, which is a Bombaceae. But among those, and on the very disturbed areas, you find a diversity of wild tomato species, and one of them is Solanum peruvianum, is one of the most widely distributed, and interestingly occurs on elevation gradients from 800 meters above sea level to close to 3,800, so close to 4,000 meters, continuous population. This is something very unusual. But what's important about this plant is that it has actually a diverse arthropod community interacting with it. So it's a diverse community of herbivores. And what we initially found was that in response to herbivory, there is herbivore-induced secondary metabolite production, so the thing that I told you about, the plants change their metabolism in response to damage. These are just different compounds that do that. Importantly, that results in an increased resistance of the plant, we call that induced resistance. So when the plant is damaged, it's more resistant than it was before. Now, interestingly, that turns out to be a problem for the pollinators interacting with the plant. And this was a surprise to us that the pollinator diversity was relatively high on those plants. And I say that's surprising because on solanaceous plants, or in particular on Solanum, the flowers have to be bus-pollinated, so vibratile-pollinated, some people say. That means that the bee has to cling onto the flower and then vibrate their wings. The pollen shoots out and they collect it and fly away. Now it turns out, because bees are herbivores too, right? They consume plant products, and in this case actually plant tissue, because Solanum flowers don't have nectar, they only provide pollen as a reward. So it turns out that when the herbivores are feeding on the plant, changing the metabolism of the entire plant, flower chemistry is changing as well, and in particular the volatile emission of the flowers. So what happens then is when you have herbivory, even a little bit herbivory, so I'm talking about in our experiments with less than 10% of the leaf tissue removed, this relatively little herbivory, the metabolism is changing, volatile emission of the flower is changing, and interestingly, this is one of those examples where suddenly the flower shifts out of the search pattern of the bees. And the bees don't perceive those flowers as hosts anymore. And this has to do with the constancy of bees. So bees learn to associate a particular signal with food. And if that suddenly disappears, it shifts out of that framework. And we called that effect herbivore-induced pollinator limitation because this is what it resulted in. It resulted in pollinator limitation, lower seed set of the plants, and so on. However, it turned out, so we did a model on this thing, and this is now the bigger question associated with that. Does that have an effect on community dynamics? So we haven't really measured that, but we did a very intense model on it that together with Mr. Clown here, who that we published in Nature Communication. And what he found was that the fact that the plant is sharing the information about the damage via producing the volatile emission is stabilizing the dynamics of all of the organisms involved in that interaction. And I just leave that with that, so you should read the paper. But this is one of those big deals. So we did pretty much the model without information and with information, with chemical information present. Without information, you get huge cycling of all of those, and you get kind of regular predator-prey kind of cycles, right? But if you add information to it, so the organisms know about it, in other words, right? It stabilizes those cycles. So communities seem to become more stable with exchange of information. That also suggests that different habitats, meaning different communities, may select for different chemical information, or like in this nice example by Andrea Kessler, who did her PhD in Lee Dyer's lab. And I just want to kind of present her example here. So she was working on a Piper Kellelii in Ecuador along an elevational gradient and looked at herbivory by AOIs. These are geometric, more or less specialized herbivores on those plants. And she looked at the chemistry, and in this case, there was resistance-mediating chemistry where a number of pyrinated benzoic acid derivatives, like this bromine here, that explain resistance. They had that shown in a previous study. But what's important, what they found was, first they did this kind of interaction network with some really interesting things here. So this is a path analysis where, interestingly, increased phytochemical defense, there was increased production of those free compounds that they had identified as particularly defense-related. Not surprisingly, decreased AOS diversity, so fewer species could share into that circle, right, of the plant, the higher the defense of the plant was. Interestingly, and this was almost like kind of counterintuitive, the parasitoid diversity went up. So meaning, this is the parasitoids, obviously, of the caterpillars here, right? Those went up, and therefore, both direct and indirect defenses are kind of upregulated the higher you are with defenses. Another interesting thing is that the diversity of pipers in that particular habitat, right? So this is the piper community diversity, had a negative effect of phytochemical chemistry, meaning the more species you had, the less chemistry you need, suggesting that there is some associational resistance kinds of effects that play a role here, right? And even though they didn't measure selection directly, this study actually supported a number of major predictions of the geographic mosaic hypothesis, namely that the relationship between interacting organisms as well as the environmental circumstances in which the interactions are played out vary on a local and regional level. And that means that they affect the strength and outcome of a co-evolutionary process. And just think that through. And this is one of those very nice examples where they used genetic tools that are now much more readily available to go through and identify the species or characterize the species. Now I'm giving you a non-tropical example. So this is goldenrod. For those of you who are in the Northeast, this is one of the dominating plant species in old field communities. Is really dominant in these old fields with covering often 75% of the entire community, and they're very stable communities, in fact. And this plant is associated with an unusually high diversity for the temperate zone of arthropods. So there are more than 100 herbivore species that are directly associated with that plant, and 85% of those are specialists. But one of them is particularly damaging, and this is that's why they call it the goldenrod leaf beetle. It's Triraptor virgata. That is in fact thriving or is the major factor of natural selection in that system, as we found out. And I just want to give you a few nice examples. So one hand, this plant again has very strong inducible resistance. So in response to damage, you have a strong induced secondary metabolism. In particular, and I tell you that specifically because it's important to understand the next slides, we have very strong inducibility of volatile emissions. So the plant smells completely different when it's damaged by a herbivore. And you can, based on the volatile emission, differentiate between two different chrysomelid beetle species on the plant. So it's highly specific, highly strongly inducible. And so there's a high induction specificity. Now it turns out the plants communicate with each other. So that means when a plant gets damaged, that volatile emission functions as information that's transmitted to a neighboring plant, which in turn induces resistance. We call that plant-to-plant communication. And initially, there were a lot of hypotheses that this is simply eavesdropping on a signal that a plant is producing simply because it has to change its metabolism to adjust to that attack, or, for example, to attract predators to the plant to kill the herbivore off, and things like that, so indirect resistance. However, in this system, things seem to be slightly different. And we took an approach where we measured the response to natural selection. And we used an experiment that Dick Root had set up while he was still alive. And we had continued, so Anurag Agrawal and I teach field ecology pretty much on the OTS model. And we had set up field plots that resembled Dick Root's old experiment, and very famous experiment, where he simply sprayed insecticide in one set of plots to exclude all insect herbivory and left others to ambient herbivory. So herbivore plus and herbivore minus means no herbivores and herbivores. And he maintained that, and he was looking at it from a community ecology perspective. So obviously different communities established here. It turned out that the dominating role that Solidago altissima has, that plant that I told you, so that tall goldenrod, is dampened by the herbivores in the system. So herbivory is increasing diversity in the community, right? Now we had the question, if it's true that different communities can select for different chemical information, then what would we expect when we exclude herbivory on a population level? So we focused only on tall goldenrod Solidago altissima and collected the plants after we had run this experiment for 12 years. We collected plants out of the plots without herbivory and with herbivory, and then set up new clone gardens, clonally propagated them, and then asked what's different about those populations. And a lot of things were different. So we had a couple of papers. Resistance was very different. As you can imagine, with herbivores, you have higher resistance traits. But what was more interesting was, without herbivores, you got very strong selection for increased competitive ability. And this is not only being better in crowing in competition, which is true, but they also produced much higher amounts, and I'm talking really 2.5 times up to 5 times higher amounts of allelopathic compounds. And these are these super reactive polyacetylene compounds that have these triple bonds. If you have a similar molar amount as Roundup and you spray that in your garden, it will kill the plants more efficiently than Roundup. So this is super highly potent competition mediating compounds, right? Allelopathic compounds. So those were the things that we had initially found. But now we looked at the volatile emission, obviously, and the hypothesis was, the question was, is communication, chemical communication between the plants actually different? And this is the best figure that was ever produced in my lab. It looks like nothing, but it is quite a bit. So what we are looking at is plant-plant communication, so the ability of a neighboring plant to perceive the information from another plant, the transmission of that information, and inducing resistance in response to that transmission, in response to the volatile signal from a related neighbor and in fact this is the same clone, so clonal plant, or from a different genotype, right? And when you look first here, this is what you expected from all the studies that we know so far in plant-plant communication, and primarily this is work by Rick Carbon. Plants only respond to their own signal. This kind of led to the hypothesis it's eavesdropping on a neighbor's signal. They only respond to the signal from themselves or from a clone of themselves, right, from the same genotype, but not to non-self. This is in the population without herbivory, that evolved without herbivory for 12 years. But here, this is the important thing. In this case, in the population with herbivory, it doesn't matter what your neighbor is, they respond equally well to the neighbor's signal within that population. So this experiment shows that there's natural selection on plant-plant communication, on induced resistance, and this is here, the structure of the chemical signal that is actually produced. And this is this is the second best figure. Because what that does is, so again, focus first on the herbivore minus, so the population without herbivores. So in both cases we see, so what this is actually reflecting is a multidimensional space, a volatile emission. Each compound in a multidimensional space gets one dimension, right? And we just press that down into two dimensions, right? And so what this is pretty much the defined information space of the population of plants that come from populations without herbivory, that evolved without herbivory and were not damaged in the experiment. This is that same population of plants, but damaged with herbivores. So what that means is that shift here, and this is pretty much the mean plus minus standard deviation. So it significantly induced volatile emission. But the size of the circle stays the same, which means your phenotypic diversity in the chemical information space stays the same, but it just shifts somewhere else. Here, in the plants, so this is the red ones, are the plants that were on the selection with herbivores, or selection by herbivores in this case. The information space also shifts, right? We have a significant induction of volatile emission, but the space becomes significantly smaller. So they converge on the same language, right? While they're all quagmiring around with different languages and change their language in response to the theory, those actually converge on the same language, which means it makes the transmission of information much more efficient, much more prevalent, right? Okay. I saved those conclusions because I kind of went through them already, and I want to get to another story very quickly. And this is the question, and this is where I see actually one of the big growing fields in chemical ecology and in tropical chemical ecology in particular, and this is the question of, can we use those information theory-driven kind of results from plant-insect interaction to control pests in our agricultural fields? And there are different possibilities there, so we can use reduction of standing herbivore populations by predation. We can change the phenotypic diversity in the plant population and thereby confuse the herbivores. And we can have species and genotype diversity that we alter also to confuse the herbivores in the field. And these are all things that have been tried. And it turns out those things that were successful either worked perfectly well in the greenhouse, and so they're used in the Netherlands, for example, in greenhouses. They use a lot of spider mites, in the US now too. Predatory mites to control spider mites, for example, parasitoid wasps to control aphids. In greenhouses, in a closed system, works perfectly well. If you go out into the field, it doesn't work anymore. The only good examples where it works in the field come from tropical systems. One is in Zaire on controlling green spider mite on cassava, where an increased volatile emission in response to herbivory of cassava, particular cassava genotypes, increases the third trophic level control of the spider mites. And the example that I want to give you here that comes from Kenya. And this is what's called push and pull, and some on the board already heard my speech on that last night. The Kenyan government charged the International Center for Insect Ecology and Physiology, Physiology and Ecology, ICIPE, with coming up with a system that biocontrols the major pests in corn and sorghum, which are the major crops in Kenya. The major pests is actually a group of stem-boring insects. I show you only one of them. This is the spotted stalk borer, Chilopartellus. So those lay their eggs on the plant. The larvae start feeding on the leaf, then drill into the stem, hollow out the stem, and at a particular point, wind goes through and the stem kinks, and you have no harvest. They can have effects of reducing harvest by up to 75% in the field. So it was a big, big problem. And ICIPE came up and applied community and chemical ecology principles to come up with a very particular system. And the people who did that were Seow Khan and John Pickett. John Pickett from Rothamsted Research did the chemical ecology of it, and Seow Khan pretty much did all the agroecology, and he was actually suggested for the food prize, World Food Prize, right? And in that year where he was supposed to get the World Food Prize, and actually New York Times already had reported that he got the World Food Prize, Monsanto got it. Do you remember when Monsanto got the World Food Prize and it was this terrible thing? But the other thing that was important in that system was that they included the farmers from the very beginning, even defining what plants they actually had in the experiment to test for the intercropping system that I will explain you in the next picture. So this is what push-pull is. So the idea is that you intercrop your crop, so this is maize here, you intercrop it with a plant that is repellent to the major pest, the push plant, and you surround the field with a plant that is much more attractive than the corn, the pull plant. And the combination of the two are so successful that it blows your mind. So they went from starting it in 2000 approximately to now having 175,000 farmers affecting the lives of 3 million people using that system. The initial kind of idea behind that, and there was quite some evidence about that. Oh, I have to show you this picture. One of the first adopters of the push-pull technology was Sarah Obama, the adoptive grandmother of Barack Obama. This is me here. We met her in 2012. Could shake her hand. Very impressive woman. So the hypothesis was, and there was some support for that, that the push plants produce volatile compounds that corn only produces when it's damaged. The stem-boring insects have a disadvantage when they lay their eggs on an already damaged plant because it may die before their offspring survives. So they tend to avoid plants that are damaged for oviposition. So that means the herbivores fly into that field, perceive the entire field as damaged, and then find an alternative host which is surrounding the field, and this was kind of driven by attractive volatiles. However, this was never put fully together. Was all kind of pieces of evidence put together. And there had to be something more to it because it worked so well in so many different habitats. And then there came Daniel Mutyambai into my lab. He is from Kenya. He did his PhD in the push-pull system. And we kind of came up with additional hypotheses for why that push-pull system works so well. There could be some plant-plant communication involved, right? So if, for example, the plant surrounding the field is inducing volatile emission that could then induce resistance in the corn plants, that would increase the resistance almost directly, right? A second thing was that different plant community effects like soil microbes and those and the crop plant chemistry could affect the chemistry of the crop plants, or maize in this case, directly. One of the most common push plants, so the intercrop, that is used is a legume in the Desmodium genus, and it actually comes from Central America. So there's Desmodium, and the root chemistry might actually induce responses in maize or sorghum because they produce these trigalactones and flavonoids as coming out of their roots, and make that fast. And I give you just one of those hypotheses, and this is this. So I'm just describing very quickly that experiment, very quickly. So this is a push-pull field, this is a non-push-pull field. So you have some weeds here, you have very little weeds, but then you have here this Desmodium. If you just take the soil out of those two fields, so we collected soil, put them into pots, put maize seeds in it, and let the maize plants grow, you get dramatic growth differences to begin with, which is primarily due to the fixation of nitrogen, probably, right? We don't know that for sure, but it is. But it turns out, and this is the big deal here, that the chemistry of the plant changes to an extent that we normally only see when herbivores are feeding on the plant. So these are just volatile emission from in green push-pull soil-grown plants and conventional soil-grown plants from the same farm. So these are pairwise comparisons that go into this comparison. Normally you see differences like this only when you have damage on the plant and you get induced volatile emission. So these plants are already in an alert state, smell damaged, and this is without any of the companion plants present, right? And overall chemistry, so this is actually an HPLC chromatogram, so these are non-volatile compounds, is also changing dramatically, including the production of the really defensive compounds in corn. And this results, interestingly, and this is now the leaf area consumed by larvae that you experimentally put on the plants, in a much higher resistance of push-pull plants. They get much less damage than the plants in a non-push-pull that come from non-push-pull soil. This is just the soil. And I leave open what that is. So we right now look into both things, microbial community changes, which is dramatically different. So we already know the microbial community is dramatically different in the soil, which may induce responses in the plants that do that, so pretty much priming for resistance. But the other interesting thing is that a lot of the chemistry from the companion plants are in the soil and may be taken up by the plant. We may have a case where the chemistry of the crop plant, in this case maize, is changing so that the herbivores can actually not perceive it as a suitable host. And with that kind of, I switch into the conclusion. So while the tropics will continue to be the major source of new discoveries of useful natural product chemistry, so this is bioprospecting, with new genetic and chemical analytical tools, the tropics will become the playing field to address major questions in chemicology, community ecology, and landscape ecology, as well as the major source of new questions in ecology and evolution of chemical signaling and communication, which is an important point, right? It's the resetting of research cycle is generating those new questions coming out of the results that you produce. And then, right now, the two best functioning applications of chemicology in the field come from the tropics. And they are a big example for the world. So tropical agriculture already provides the best example for sustainable application of chemical and community ecology and will become the global model for it. So this is where the big future lies, actually, I believe. And with that, I'm stopping here with a nice view.